This is nature. But what happens when the natural order is disturbed? I am Nicholas Blake, and I will take you on a journey exploring human reproductive cloning.
A gene is a unit of heredity found in a small sequence of DNA inside a chromosome. In all nucleic cells in our body, there are chromosomes, which are tightly wound genes and store incredible amounts of data. Inside each cell in our body, we have 23 pairs of chromosomes, which contain the blueprints for the formation of everything in our body. Now, the most obvious exception to this is gametes, or our sex cells. Our gametes, or our sex cells, contain only 23 chromosomes. This is because when the sperm and the egg combine, the usual 46 or 23 pairs of chromosomes has been attained, and enough information is then given to begin the reproduction. Now, the combination of maternal and paternal genes, or our mother and father's genes, provides us with the genetic diversity that we see all around us each day.
In reproductive cloning, the nucleus of an egg cell is removed, and the nucleus of a somatic cell, usually skin cell, is planted into the egg cell. This process is called somatic cell nuclear transfer, or most commonly referred to as SCNT. With the SCNT method, the somatic cell that is taken is that of the person who is being cloned. It is placed in the egg and is manipulated to divide into an embryo using chemical or electrical stimulation. After a few days, the embryo will have developed enough to be put back into the mother. It is important to note, however, with reproductive cloning, that the offspring will not be considered a mother, father, son, or daughter. It will be considered a clone. The idea of the SCNT method had been around for some time, but it wasn't until 1928 when Hans Spemann, a promising German scientist, put the hypothesis to the test. He successfully transferred a salamander skin cell into an egg cell, but he was not able to stimulate embryonic growth chemically or electrically. In 1952, the work was continued by Robert Briggs and Thomas J. King. Their work specified in amphibious cell nuclei, specifically those from tadpoles. By the end of their work, the two men had successfully cloned 27 tadpoles and 35 embryos from their 104 nuclear transfer attempts. It was their work that inspired decades of research and investigation into reproductive cloning.
John Gurdon of Oxford University discovered in 1962 that stem cells could be used as an alternative to somatic cells. Now, he discovered that when these stem cells activated as somatic cells, or our skin cells, that no genetic information was lost. Whilst this attracted the attention of many, other scientists did not believe that Gurdon's work was legitimate, finding many flaws.
Research and development continued into nuclear transfer work, and the use of mammal cells became the new craze amongst the scientific field. This resulted in the conception of the first mammal cloned from an adult nucleus taking place in 1996 by Ian Wilmot and Keith Campbell of the Roslin Institute in Scotland. The birth of the clone, Dolly the sheep, was first announced in the journal Nature in 1997 and initiated the worldwide discussion about the possibility of cloning humans. As demonstrated by this image, it is known that Dolly the sheep was cloned using the somatic cell nuclear transfer method.
Following Dolly the sheep, many other animals had been attempted to be cloned by other scientists. These include horses, mice, sheep, salamander, cats, and more salamander. The results of these experiments all point to one thing. Skunt is possible for human cloning.
The discovery of this technology and the understanding of human cloning is undoubtedly a significant contribution to modern science. But the question remains whether human cloning is a good idea and is a topic that needs much discussion and speculation. Despite this widespread opposition to reproductive cloning, some scientists have nonetheless pursued this goal, and several claim to have successfully cloned humans and even to have brought some to term. The first human clone, a healthy 7-pound girl named Eve, was born on the 26th of December 2002, according to CloneAid, a private company linked to the Raelian religious group. The Raelians believe that human life was created in an alien laboratory and that human cloning provides the opportunity for human immortality. The claim was never verified. In 2003, a professor from the University of Kentucky claims to have cloned a human embryo but to have cryopreserved it at an early stage of development. In November 2002, Dr. Severino Antonori announced that he had successfully used cloning to induce pregnancy in 3 women, with the birth of the first child expected in January of 2003. However, none of these claims have been found to be true or have been verified.
Reproductive cloning is a very delicate topic in our modern society, as it has many ethical issues that are constantly debated upon by our population. Now we will discuss the pros and the cons.
Many people of the scientific community believe that reproductive cloning could serve as an alternative to natural conception for those who are unable to birth a child. They also say that this form of human cloning could allow for an easier supply of donated organs, if scientists were able to specifically clone such organs. However, there are a large number of people who strongly disagree with human cloning, and say it wipes all individuality away from our race. They say that cloning objectifies humans, and classifies the clone as the possession or belonging of another. Under the direction of the United Nations, reproductive cloning of humans had been outlawed in most countries due to ethical, moral, and medical reasons. Based on this information, we can say that there will be no effect on society or on any demographic, as cloning is not permitted. However, there is more to the story. Current law states that a cloned embryo may not be brought to term or developed past embryonic state, so scientists are still free to practice the skunt method as long as they don't continue past an embryo. The skunt method is still being used to research therapeutic cloning, and in the eyes of certain groups such as the Catholic Church, this is seen as a disgrace to human nature and an upset of the natural order. If humans were able to clone to full term and give birth to a clone, there would be shattering effects on religious societies. Catholic rule outlines that the process of making a human being is a sacred and passionate procedure carried out by 2 human beings, and that a person's uniqueness is something held very dearly, and cloning directly contradicts this. So folks, that concludes the end of our program. We hope you enjoyed. Before you go, we'd like to ask you a few questions.
What do you, as the viewer, believe about reproductive cloning? Are you for or are you against? Leave a comment down below.
Thanks for watching, folks. Till next time.